Hi class, this is your instructor, Skylar Huff. We're in chapter 31. This is fungi. Fungi are found everywhere on Earth, from the tropics to the tundra, and in both terrestrial and aquatic environments, and have a profound influence on ecology and human health. Although they are often thought to be closely related to plants, fungi share a more recent common ancestor with you and me, animals, and are therefore more closely related to animals than they are to plants. This can cause significant problems for the treatment of fungal pathogens of animals. The two groups' physiological similarities mean that drugs that can kill fungal pathogens may also harm host animals, including humans. Fungi secrete digestive enzymes and then absorb the externally digested nutrients that are rapidly distributed throughout the organism. This allows for rapid growth and adaptation to a wide range of ecological niches. Fungi made it possible for plants to colonize land by associating with rootless stems of plants to aid in the uptake of nutrients and water. Although some fungi cause disease in plants, they play critical roles in the carbon cycle, breaking down dead and dying plant matter to rapidly mobilize plant fixed carbon into forms of carbon usable by other organisms. Some fungi, like the fly agaric pictured here, produce toxins, but other fungi are used as food by humans, and yeasts have been used for millennia to make breads and alcoholic drinks. Early attempts at classifying fungi relied on comparisons using their gross morphological features. Advances in microscopy and biochemistry in the late 18th, 19th, and 20th centuries allowed refinement of earlier classifications. Then, rapid advances in molecular biology in the 1980s allowed for more accurate classifications that better reflected the evolutionary relationships between different groups of fungi. Modern molecular techniques, including genomics, are revealing the unique evolutionary journey that has shaped this kingdom and will continue to inform new classifications and approaches to systematics. Phylogenetic analyses of DNA and protein sequences show that fungi are more closely related to animals than to plants. Fossils and molecular data indicate that animals and fungi diverged about 1.3 billion years ago. Their common ancestor was unicellular, and from that point, unique solutions to multicellularity evolved in both fungi and animals. Attempts to resolve the phylogenetic relationships between fungal groups have stimulated much debate. Molecular data can be used to create classifications based on alternative hypotheses. Traditionally, 4 fungal phyla were recognized, based primarily on characteristics of cells undergoing meiosis. Chytridiomycota, the chytrids, Zygomycota, Zygomycetes, Ascomycota, the Ascomycetes, and Basidiomycota, the Basidiomycetes. To traditional classification. The discovery that Zygomycota and Chytridiomycota were paraphyletic led to the subdivision of the chytrids into 3 distinct phyla: Chytridiomycota, Blastocladiomycota, and Neocalamystigomycota. Similarly, the fungi traditionally grouped in the Zygomycota were closely related to based on the fact that they share a reproductive structure called a zygospore. With more data on molecular and genetic comparisons, these traditional Zygomycetes are in the process of being reclassified into multiple phyla. There are now 6 widely recognized monophyletic groups that make up kingdom fungi: Blastocladiomycota, Neocalamystigomycota, Chytridiomycota, Glomerulomycotina, Basidiomycota, and Ascomycota. Although the relationships within the paraphytic zygomycota have yet to be resolved, most classifications of kingdom fungi also include this group. Another group, Microsporidia, have often been studied by mycologists despite not being considered true fungi. Recent molecular evidence suggests that they are closely related to some fungi of the zygomycetes and should be classified at least as a sister group to fungi. A relatively new group, Cryptomycota, has been suggested as a seventh phylum. This group includes a diverse yet uncultured group of fungi, so far only detected by molecular techniques in soil and in marine and aquatic environments, confounding our understanding of the complex evolutionary relationships between fungi. Is a growing body of evidence that horizontal gene transfer has occurred between many fungal species. We usually become aware of fungi when we see them growing in our lawns, on trees, or on the bread left out on the counter too long. However, mycologists, scientists who study fungus, estimate that there are somewhere between 2.2 and 3.8 million species of fungi. Although only about 5% have currently been classified. Fungi exist as either unicellular organisms called yeasts or in multicellular form. Fungi use one life strategy or a combination of them to succeed in varied ecological niches. Some are mutualistic, some are parasitic, and some are free-living. Their reproduction may be either sexual or asexual, and they exhibit an unusual form of mitosis. They are specialized to absorb nutrients from their surroundings by secreting digestive enzymes. Other major features of fungi include a cell wall composed of chitin and reproduction by spores. Fungi may be the Chytridiomycota and Microsporidia exhibit many ancestral traits that are exclusively unicellular. During the evolution of the higher fungi, filamentous forms developed. Fungi grow in two basic ways, as unicellular forms called yeasts and masses of multicellular thread-like forms called mycelia. Mycelia are aggregations of slender filamentous structures called hyphae. Hyphae consists of single cells joined end to end, surrounded by a common cell wall. Individual cells can be separated from each other by a perforated cell wall called a septum, which allows the flow of the cytoplasm, making them effectively one large cell. The streaming of cytoplasm from cell to cell in a hypha is an adaptation that allows fungi to grow rapidly when nutrients and moisture are abundant. The molecules needed to build cellular components can quickly be moved to the growing tips of hyphae, allowing very rapid growth rates. You may even have seen mushrooms suddenly appear overnight in a lawn after a summer rain. Some types of fungi have hyphae that lack septa entirely and are one giant cell containing multiple nuclei. These structures are called coenocytic hyphae. A mass of connected hyphae is called a mycelium. A mycelium is the main body of multicellular fungi that grows into, on, or through soil, wood, or tissues of host organisms. Mycelia can be huge, with hyphae collectively being many meters in length, such that mycelia cover acres of ground. Because of the thread-like nature of hyphae, a mycelium has a high surface-to-area volume ratio. This means that the absorption of nutrients is very efficient. This also contributes to the high growth rates of some fungi under ideal conditions. When resources become scarce, sexual reproduction is favored to introduce new variants. When facing such conditions, spore-producing structures called fruiting bodies can form rapidly from dense masses of hyphae. The spores produced by these structures are resistant to adverse conditions, such as desiccation, and germinate when conditions become suitable. These reproductive structures are most obvious in the forms of mushrooms, toadstools, and puff balls. Unlike cell walls of plants and many protists, fungi have cell walls composed primarily of the polysaccharide chitin. Like the cellulose found in plant cell walls, chitin is a polymer. However, chitin is made from a modified nitrogen-containing form of glucose called N-acetylglucosamine. The polymers of N-acetylglucosamine are then cross-linked with proteins. Chitin is also found in the exoskeletons of mollusks, insects, and crustaceans. The presence of cell walls made of chitin in both animals and fungi is part of the evidence supporting the idea that fungi are more closely related to animals than they are to plants. In addition, fungi and animals also both store excess glucose as glycogen, while plants store glucose as starch. Fungi are different from most animals and plants in that each cell or hypha can house two or more nuclei. A hypha that has only one nucleus is monokaryotic. A cell with two nuclei is dikaryotic. In a dikaryotic cell, the two haploid nuclei exist independently. Dikaryotic hyphae have some of the genetic properties of diploids because both genomes may be transcribed. Sometimes, many nuclei are found in the common cytoplasm of a fungal mycelium, which can lack distinct cells. If a dikaryotic or multinucleate hypha has nuclei that are derived from two genetically distinct individuals, the hypha is heterokaryotic. Hypha that have two or more genetically identical nuclei are called homokaryotic. The reproduction of fungi is dramatically impacted by both the organization of hyphae and the way in which tissues are sometimes organized to produce unusual body designs. These features create some unusual reproductive strategies that include, in some species, the ability to reproduce both sexually and asexually. These strategies have likely contributed to both the diversity of fungi and their success in a range of ecosystems, including those where there is significant variation in conditions. Some reproductive strategies allow fungi to survive conditions that are not conducive to growth. Most higher eukaryotic cells are characterized by replication and segregation of the genetic material, followed by a division of the cytoplasm. This makes the cell unit of division. In many filamentous fungi, however, the nucleus is the unit of division, and cytokinesis does not occur. In many fungal mitosis, the nuclear envelope does not break down as it does in higher eukaryotes. And the spindle apparatus with the nucleus, except for the chytrids, centrioles are absent in fungi. Many fungi are capable of making both sexual and asexual spores. When a fungus reproduces sexually, two haploid hyphae of compatible mating types may come together. Because of specific mechanisms for reproduction are often specific to and are sometimes defining features of the main groups of fungi. The details of reproductive and life strategies are discussed in detail in just a few. Some fungi produce specialized reproductive structures to make spores. For example, the mushrooms and puffballs can be seen above ground, and the shelf fungus appears on the trunks of trees or small spore-forming structures. These structures are made from dense colonies of hyphae that arise from a mycelium. Unlike in a mycelium, the hyphae forming the reproductive structures have cells separated by intact septa, and there is no cytoplasmic streaming. There are four general groups of reproductive structures formed by fungi: modal zoospores, zygosporangia, basidia, and asci. Spores are the most common means of reproduction among fungi, and to varying degrees, they are resistant to drying out, heating, and freezing. They may form as a result of either sexual or asexual processes, and they are often dispersed by wind, insects, or small animals. Some spores have adaptations so that they can serve as vehicles for distributing the parental fungal genome to a more favorable location when conditions become adverse. Other spores have structures more consistent with providing long-term protection for the parental genome. They have cell walls highly resistant to desiccation and are nutrient dense. These spores have evolved to persist in the environment, providing the protection and means for waiting until such conditions become favorable or new nutrients appear in the environment. Fungi, like animals, are heterotrophs. They obtain reduced carbon and energy from the digestion of complex polymers. However, animals first ingest their food, bringing it into their bodies, before using hydrolytic enzymes to break down the polymers inside specific digestive organs. Proteins are broken down into amino acids in our stomachs and small intestine. Fungi, by contrast, secrete digestive enzymes into the environment where the polymers are broken down into smaller molecules. The fungi subsequently absorb the digested organic molecules. In fungi that act as decomposers, the material may be degraded is commonly dead plants and animals on which the fungus can live. However, parasitic fungi will invade the living cells of their hosts and absorb nutrients directly. The structure of fungi reflects their nutritional strategy. Unicellular fungi, yeasts, have the greatest surface area to volume ratio of any fungus, maximizing the surface area for nutrient absorption. The extensive thread-like networks of hyphae and mycelia also provide an enormous surface area for enzyme secretion and nutrient absorption. Many fungi are able to break down cellulose in wood, cleaving the bonds between glucose subunits, and then absorbing the glucose molecules. Most fungi also digest lignin, an insoluble organic compound that strengthens plant cell walls and is found in wood and bark. The specialized metabolic pathways of fungi allow them to obtain nutrients from woody plants and from an extraordinarily large range of sources, including tiny roundworms called nematodes. In the environment, the majority of dead plants and plant-based materials are decomposed by fungi. Without this activity, ecosystems will become overwhelmed with dead plant material. Fungi, along with bacteria, are the principal decomposers in the biosphere. They break down organic materials and return the substances locked in those molecules to circulation in the ecosystem. Fungi can even break down cellulose and lignin, an insoluble organic compound that is a major component of wood. By breaking down the macromolecules in dead organisms and feces, fungi release carbon, nitrogen, phosphorus, and make them available for other organisms. Because of their ability to break down almost any carbon-containing compound, fungi have potential uses in bioremediation. For example, they have the potential to clean up forest litter, soil, or water that's contaminated. Although fungi, like bacteria, are somewhat innocuous in our day-to-day lives, their roles in ecosystems and diverse symbiosis they form make them critical in the functioning of the biosphere. Climate changes directly affect fungal distributions and diversity, which in turn affect nutrient cycling. biogeochemical cycles, and ecological energetics. Moreover, changes in animal and plant distribution and diversity due to climate change will likely have significant indirect impacts on fungal symbiosis and subsequently ecosystems biology. Endophytic fungi. Some species of fungi live inside plants, residing in intercellular and apoplastic spaces, obtaining nutrients from the plant. In some cases, fungi produce compounds such as alkaloids that protect the plant against herbivory. In other cases, fungal products might increase plant nutrient absorption by that plant. Lichens are symbiotic associations between a fungus and a photosynthetic partner. Lichens tend to benefit both partners. The photosynthetic partner provides carbohydrates to the fungus, while the fungi provides moisture and nutrients for the algae or cyanobacteria. Ascomycetes are the fungal partners in all but about 20 of the approximately 15,000 species of lichens estimated to exist. Most of the visible body of a lichen consists of its fungus. But between the filaments of that fungus are cyanobacteria, blue-green algae, or sometimes both blue-green algae and cyanobacteria. Although the main body, the thallus, of a lichen can spread to be many centimeters across, the absence of water-conducting structures means that lichens cannot grow vertically and thus cannot compete with plants for light. Special fungal hyphae penetrate or envelope the photosynthetic cell walls within the photosynthetic partner and transfer nutrients directly to the fungal partner. Although fungi penetrate the cell wall, they do not penetrate the plasma membrane. Biochemical signals are sent out by the fungus, apparently direct its cyanobacterial or green algal component to produce metabolic substances that it does not produce when growing independently of the fungus. The fungi and lichens are unable to grow without their photosynthetic partners. The fungi protect their partners from strong light and desiccation. When fungi components of lichens are isolated from their photosynthetic partners, they survive but grow very slowly. Here are lichens. And before I go too far, I should mention the durable construction of the fungus combined with the photosynthetic properties of its partner allow lichens to grow anywhere it is relatively dry with a stable substrate for growth. Lichens are resilient to drying and can survive extended periods without water, remaining capable of growth if provided with moisture. In harsh exposed areas, lichens are often the first colonists, breaking down rock to produce new soil and setting the stage for colonization by other organisms. Given their adaptations to extremes of temperature and their resistance to desiccation, lichens can be more successful than plants in some ecosystems. About 90% of all plant families have species with roots that form obligate symbiotic relationships with fungi called mycorrhizae. These symbioses are so pervasive in plants that these fungi probably amount to about 15% of the total weight of the world's plant roots. The fungi and mycorrhizal associations act as extensions of the plant root system. The fungal hyphae dramatically increase the surface area of the plant root in contact with the soil, resulting in much more efficient absorption of nutrients such as nitrogen, phosphorus, and trace elements. The plant, on the other hand, supplies sugars to the fungus, so symbiosis is mutualistic. Not only do mycorrhizal fungi play an important role in the health of plants with which they associate, they also play a critical role in maintaining soil moisture. For example, many mycorrhizal fungi secrete glycoproteins into the soil that promote the adherence of soil particles, which contributes to soil stability. In turn, this promotes the health of all plants in the soil and can reduce soil erosion. The contributions of mycorrhizal fungi, along with other soil fungi, to the global carbon cycle is also important. The soil carbon pool is roughly 3 times as large as atmospheric CO2 carbon pool. There are 2 types of mycorrhizae, arbuscular mycorrhizae and ectomycorrhizae. Arbuscular mycorrhizae have hyphae that penetrate the outer cells of the plant root. They grow through the plant cell walls and make contact with the plant root cells plasma membrane via highly specialized branch structures called arbuscules. Ectomycorrhizae have hyphae that grow around and surround but do not penetrate the cell walls of root cells. In both cases, the mycelium extends far out into the soil, so the fungus is interacting more closely with the soil via its thread-like hyphae than can root hairs. A single root may associate with many fungal species, creating a complex set of symbiotic relationships across species and kingdoms. Arbuscular mycorrhizae are far more common than ectomycorrhizae and form roughly 80% of all plant species. The fungal partner in the arbuscular mycorrhizae is always a member of the subphylum Glomeromycotina, which is why these fungi are called AM fungi. The AM fungi also associate with more than 200,000 species of plants and are likely the major route by which plants obtain inorganic soil nutrients. Arbuscular mycorrhizae. The majority of fungal species in ectomycorrhizae are basidiomycetes. Unlike the arbuscular mycorrhizae, these species often produce above-ground reproductive structures. Such as mushrooms. Excuse me. Ectomycorrhizae are associated with significantly fewer plant species than are arbuscular mycorrhizae. However, this should not be taken to mean that their role in the ecosystem is less important than that of arbuscular mycorrhizae. Many of the ectomycorrhizae hosts are tree species that are significant carbon sinks for the atmosphere, such as pines, oaks, and birches, which are also important timber species. Ectomycorrhizae. Numerous fungus-animal symbioses have been identified. For example, ruminant animals contain certain fungi in their gut that can release nutrients by degrading plant materials with high cellulose and lignin content. In return, the fungus gets to live in the nutrient-rich rumen of the host animal. A notable fungus-animal symbiosis occurs between leafcutter ants and some species of basidiomycetes. These ants, members of the phylogenetic tribe Attini, have an obligate symbiosis with specific fungi, which they have domesticated and form in an underground garden. Fungi can destroy crops and create significant health problems for humans and other animals. A major problem in the treatment and prevention of animal disease caused by fungus is that, like animals, fungi are eukaryotes and share significant physiological similarities with the animals they infect. Understanding how fungi are different from plants and animals may lead to better treatments for fungal infections. Fungi can cause health issues in humans in three ways: by acting as allergens that trigger immune responses, by direct infection, usually of the skin and nails, called mycoses, or by producing secondary metabolites called mycotoxins that, if ingested, can lead to chronic or acute illness. Spores in indoor air can trigger allergic responses similar to those triggered by pollen grains in the air, producing characteristic hay fever symptoms. Fungal spores can also trigger more serious allergic reactions, such as asthma in both children and adults. There is a growing public health concern regarding high levels of spores produced by fungal growth in flooded homes that displaced families return to following natural disasters. Some spores produce only allergic responses, but some species of spores also contain high levels of mycotoxins that could lead to illness if humans come in contact with them in sufficiently high numbers, such as may be achieved in a closed environment once a flooded basement that becomes contaminated with mold. Many species of fungi produce mycotoxins. Some of these mycotoxins, such as those produced by some members of the genus Penicillin, are used to make antibiotics to treat bacterial infections. But several thousand types of fungus produce mycotoxins that can be harmful to humans. Mycotoxins are most commonly ingested when contaminated foods are eaten or when steaks are made, identifying edible raw mushrooms. Aflatoxins are a type of mycotoxin produced by Aspergillus species that grow on various staple foods such as corn, peanuts, cotton seeds, and other grains and spices. Aflatoxins can cause several kidney and nervous system damage in animals, including humans. Aflatoxin B1 is one of the most carcinogenic compounds known, being nearly 100 times more carcinogenic than cancer-causing compounds in cigarette smoke. There are also fungi that infect plants that are beneficial to humans. Corn smut is a maize fungal disease caused by the Basidiomycete Eustalago-Medes that is harmful to the plant, but not to animals that consume it as a food. Fungi can also infect humans and other animals to cause disease. Some common diseases, such as ringworm, which is not a worm, but a fungus, athlete's foot, onychomycosis, a fungal infection of the toenail, and oral and vaginal candidiasis, can be treated with topical antimycotics, and in some cases, with oral medication. However, fungal infections may be difficult to treat because of close phylogenetic relationships between fungi and animals. When a pathogen is closely related to the host, it can be difficult to find drugs that target the biology of the pathogen, but which do not interfere with the biology of the host. When two groups of organisms are well separated phylogenetically, such as bacteria and humans, it is easier to find drugs that discriminate between the biology of the two groups. The health of non-human animals can also be affected by fungi. For example, the disease Ch31 was first identified in 1998 as an emerging infectious disease of amphibians. Amphibian populations have been declining worldwide for over 3 decades. The decline correlates with the presence of the chytrid in the amphibian's skin. Sick and dead frogs were more likely than healthy frogs to have flask-like structures encased in their skin, which proved to be associated with chytrid spore production. Fungal species cause many diseases in plants and are responsible for considerable loss in food crops every year. In the past, the Irish potato famine and both Dutch elm blight and chestnut blight in America were devastating agricultural diseases caused by fungi. Today, rice blast disease and wheat stem rust poses threats to staple food crops. In addition to causing plant disease, fungi also spoil food products that have been harvested. These threats to food security are especially concerning given global population expansion, most of which happens in developing countries and are most worrisome for countries susceptible to changing climate patterns. Changes driven by a few major contributing countries, and generally not those countries that would be most adversely affected by crop loss. Many plant infections are established when a spore arrives on the plant surface. This can happen due to wind dispersal of spores, through transmission on an animal vector, via water splashing onto the plant surface, or through the soil. The basidiomycetes, following Basidiomycota, are a large group of fungi with over 32,000 known species. Along with the Ascomycota, they comprise a subkingdom within the fungi called Dicaria. Some of the most familiar edible fungi, such as portobello and button mushrooms, are basidiomycetes, but so too are many important plant pathogens, including rusts and smuts. Rust infections resemble rusting metals and smut infections appear black and powdery due to the spores. Many mushrooms are used as food, but others are hallucinogenic or highly poisonous. The famous death cap mushroom, which produces one of the most lethal toxins known, is another well-known member of Basidiomycota. There are also basidiomycetes that cause disease in animals. There is significant variation between the life cycles in basidiomycete life cycles. So the description is of the life cycle for a typical mushroom. Following germination of a haploid basidiomycete spore, a branching network of monokaryotic hyphae is formed by mitotic cell division. In the hyphae, individual genetically identical haploid nuclei are separated by the septa. Because all nuclei are genetically identical, the mycelium is homokaryotic. These hyphae form a primary mycelium. The majority of basidiomycete mycelium is below ground, where dispersal of spores would be extremely restricted. To facilitate spore dispersal, reproductive structures called basidiocarps form. These structures can be seen as the caps of mushrooms or toadstools on lawns or rotting logs. The basidiocarp formation begins with the aggregates of dikaryotic hyphae. Under correct conditions, these can develop very rapidly into above-ground structures called basidiocarps. At the tips of the hyphae gills, which are sheets of tissue on the underside of the cap of a mushroom, club-shaped structures called basidia form. In these structures, the haploid nuclei in the dikaryotic hyphae fuse in the process of karyogyny to produce the diploid nucleus. The diploid nucleus undergoes meiotic cell division to produce up to 4 haploid basidospores that are held at the end of basidia on slender stalks called sterigmata. Released basidospores are easily dispersed by wind, and under suitable conditions, they germinate and restart the life cycle. The gills on the underside of a basidiocarp increase the surface area over which spores can be dispersed by up to 20 times that which would be achieved if dispersal were performed on a flat surface. Estimates predict A mushroom can produce as many as 100 million spores per hour. And that is a death cap mushroom. The phylum Ascomycota contains over 64,000 species, comprising more than half of the known fungal species. Ascomycota is an incredibly diverse phylum, including such familiar and economically important fungi as bread yeasts, common molds, morels, cup fungi, and truffles. Also included in this group are many plant pathogens, those that cause chestnut blight and Dutch elm disease. Maybe the most famous ascomycete is penicillium, which produces penicillin. As in the Basidiomycota, there is significant variation in the life cycles of different species of ascomycetes. Like in Basidiomycota, the germination of ascospores produces monokaryotic hyphae by mitotic division, and the haploid nucleus and division of cytoplasm by septum formation. The hyphae nuclei are separated by septa. However, the cytoplasm flows through the hyphae. Asexual reproduction is common in the ascomycetes and results in production of conidia. These are the asexual spores cut off by septa at the ends of modified hyphae called conidiophores. Conidia are easily dispersed by airflow, water, or animals and allow for the rapid colonization of a new food source. So shown to you all there is a morel and a cup fungus. Unlike the Basidiomycota, the mycelia are produced by masses of ascomycete hyphae, and they remain monokaryotic or homokaryotic, and no extensive vegetative secondary mycelium forms. Sexual reproduction is initiated when two compatible hyphae make contact. One hypha produces an antheridium, the other produces an ascogonium. The resulting dikaryotic cell divides by mitosis, forming a fruiting body called an ascocarp. While the ascocarp is dikaryotic, it remains monokaryotic. So structures called asci or an ascis form on the tip of the dikaryotic hyphae. Each diploid cell undergoes meiosis to produce 4 haploid cells, which immediately undergo mitosis, one at a time, to form 8 haploid ascospores. Ascospores are the sexual spores of Ascomycota. Some ascomycetes. Most yeasts are ascomycetes with a single cell life cell. Most reproduce asexually by cell fission or budding. When a smaller cell forms a larger one, sometimes 2 cells of the opposite mating type fuse to form a dikaryon. This cell may then function as an ascus with kerygamy followed by immediate meiosis, and the resulting ascospores germinate into new asexually reproducing yeast cells. The ability of yeast to ferment carbohydrates, breaking down glucose to produce ethanol and carbon dioxide, is exploited in the production of bread, beer, and wine. Many different strains of yeast have been domesticated for and selected for these processes using sugars in rice, barley, wheat, and corn. The most important yeast in baking and brewing and winemaking is Saccharomyces cerevisiae. This yeast has been used by humans throughout recorded history. Yeast is also employed as a nutritional supplement because it contains high levels of B vitamins and because 50% of yeast is protein. Saccharomyces cerevisiae is a genetically traceable model organism long used in biological research. It was the first eukaryote to be manipulated extensively by the techniques of genetic engineering, and yeasts still play roles as models to help us better understand biology and genetics in higher eukaryotes. Glomeromycotina. Despite being a small group of fungi with only 200 or so known species, this group is important for two related reasons. First, they are obligate symbiotes with the plant roots, where they form arbuscular mycorrhizae, increasing nutrient absorption for the plant while receiving sugars in return. And secondly, due to their symbiosis with plant roots. So they were likely instrumental in colonization by land by plants. Traditional zygomycetes, the zygote-producing fungi. About 100 named species of fungi form sexual spores in Zygosporangium. Because of this shared feature, they were traditionally grouped together with Zygomycota. Asexual reproduction occurs much more frequently than sexual reproduction in zygomycetes. During asexual reproduction, hyphae produces clumps of erect stalks called sporangiophores. The tips of the sporangiophores form bulbous sporangia, and these structures are the fluffy white mold you might find growing on the surface of old bread. In conditions where sexual reproduction may not be advantageous, or asexual reproduction might not be advantageous for a fungus, chemotropic hyphae of opposite mating types attract one another. Gametangia from opposite mating types undergo plasmogamy, and the resulting fused structure becomes isolated from the parental hyphae by the formation of septa. Inside the fused gametangia, haploid nuclei undergo kerigamy to produce diploid nuclei. The fused gametangia containing diploid nuclei develop into a structure called a zygosporangium. Inside the zygosporangium, zygospores develop, producing thick coats to help the spores survive unfavorable growth conditions. Unlike the process involved in basidiospores or ascospores, meiosis to produce haploid zygospores is delayed until germination of the zygospore. So this is what's found in Rhizopus, the genus, or the scientific name Rhizopus stolonifer, for the name of black bread mold. Ch31 initially grouped together with a single paraphyletic Ch31 was recently divided by molecular analyses into 3 monophyletic phyta. I will be here very briefly. Members of the phylum Ch31, the Ch31mycetes or chytrids are aquatic flagellated fungi that closely resemble the ancestral fungi. So the production of zoospores, which are flagellated asexual spores. So, as well as being abundant in aquatic and marine environments, chytrids are also commonly found in soil. Several species are pathogenic, infecting plants, algae or other fungi, and amphibians. So this is why many species of amphibians have begun to die off over the past 30 years. And there is one of those zoospores with its flagella. We now have blastocladiomycetes, also known as blastoclads, found in both aquatic and terrestrial environments. They are parasites of plants, invertebrates, algae, and in one case, other fungi. Possibly the most distinguishing feature of the blastomycetes is a haplodiplontic life cycle, where the alternation between haploid and diploid generations in multicellular forms of the organism. Otherwise, you would have likely heard of these called water molds. And lastly, we have neocolamistomycetes. The neocolamistomycetes are fungi that can enzymatically digest cellulose and lignin of the plant biomass in the grassy diet of many ruminants. Sheep, cows, kangaroos, elephants all depend on these fungi to excuse me, to obtain sufficient nutrients and calories. The cellulose-degrading neocolamistic mycete population of ruminants is critical when these animals are fed diets that are otherwise very difficult to digest. These fungi lack true mitochondria and instead use hydronosomes, which are thought to be derived from mitochondria to produce ATP. Due to the anaerobic growth of these fungi and their abilities to degrade cellulose at relatively high temperatures, they have potential uses in biotechnology for biofuel production. This has been your instructor, Scholar Huff.